So good afternoon, everyone, and welcome to today's Biomarkers series webinar, Breath Biopsy Omni for comprehensive and reliable detection of VOC biomarkers in breath. My name is Esther, and on behalf of Oxford Global and Alstone Medical, I'd like to thank you all for joining us today. After the presentation is complete, we will have a 10-minute Q&A session. Please submit your questions through the question panel on your menu display. The full recording of this webinar will be made available to download on our secured website, and the details will be sent to you shortly after today's session. Non-invasive biomarkers are of great interest for use in disease screening and ongoing health monitoring. Researchers and clinicians seeking reliable non-invasive methods for biomarker discovery, including both untargeted and focused approaches, should join this webinar. The presentation will cover applications of non-invasive biomarkers for early detection of disease and precision medicine. Alstone Medical's breath biopsy is suited to the collection and analysis of breath samples across a range of therapeutic areas, including respiratory and inflammatory diseases, oncology, and liver disease. This webinar will present case studies from these areas and Alstone Medical's approach to developing biomarker solutions. Breath sampling is regarded favorably by patients and is suitable for ongoing monitoring and early detection, enabling faster decision-making in clinical trials. Unlike other sample matrices, large volumes of breath can be collected and concentrated, improving analytical sensitivity, and sampling is completely non-invasive. It can be performed at more frequent intervals. In this webinar, Drs. Elizabeth Crone and Tina Sho will explore the diverse clinical applications of breath biomarkers and the benefits of non-invasive breath analysis for drug development, diagnostics, and therapeutic decision-making. They will also demonstrate breath biopsy and its capabilities for early detection of disease and precision medicine through a series of case studies. Finally, they will demonstrate how breath biopsy allows robust untargeted biomarker discovery through the Omni assay and show the breath biopsy VOC atlas, a catalogue of identified and quantified volatile organic compounds, VOCs, that are commonly found on breath within a heterogeneous healthy population. Dr. Elizabeth Crone is a business development manager in Alstone, focusing on Europe and Asia. She has a scientific background in biomolecular analytics with a PhD from the University of Cambridge, looking at protein stability in conditions of oxidative stress. She has spent the last decade working for companies who have novel and interesting technologies and ideas. She specializes in helping to connect that science with the researchers who could benefit from using it. Dr. Tina Sho joined Alstone in September 2021 as a biomarker scientist, where she is involved in helping customers study design, biological results interpretation, and final report delivery. She is also involved in some of the internal R&D projects that revolve around improving the platform for breath analysis. So without further ado, over to you now, Liz and Tina.
Thanks for joining this webinar today. I'm Liz Crone, and I'm here with my colleague Tina to talk to you about how breath can be used as a biomarker matrix, focusing specifically on how it can be used to support drug development. Alstone as a company are very focused on breath as a matrix to study biology. We're looking at a range of molecules called volatile organic compounds, that's VOCs for short, which are very small molecules that reflect a variety of sources, including both human and microbial metabolism. And there are plenty of papers that have been published on these kinds of molecules linked to a range of different biological processes and diseases. So in breath, we will see VOCs that come from the lungs themselves. So from the lung tissue and the lung microbiome, for example, they'll produce VOCs that can be seen in exhaled air. But we'll also see VOCs that come from the wider body, as these molecules are really small and they readily diffuse between blood, tissue and air. That means a VOC can be produced at a distal location in the body and diffuse into the bloodstream, into the alveoli, and then can be seen in exhaled air. But why would you study these in the first place, and why would you want to look at breath for that matter at all? At Alstone, we believe there are lots of different reasons why breath, and specifically exhaled VOCs, could be interesting to look at as potential biomarkers. So firstly, breath is completely non-invasive, it's pain-free, and it's very well tolerated, which means it's very easy to get from patients. Secondly, it's a completely orthogonal approach to other biomarker platforms. VOCs don't typically get looked at in other matrices, so you can get compatible information that can help with studies. Third, breath is very accessible. It's easy to get from patients and has potential to translate to point of care type monitoring or even home-based testing in the future. As breath is completely inexhaustible, you're not going to stop breathing. You're not limited by the amount that you can actually collect, unlike with tissues and with biofluids. And combined with the fact that the VOCs that we're looking at can be concentrated from a large volume of breath into a much smaller sample, you can actually really get to very low detection limits on breath. So it's a very sensitive matrix to look at for VOCs. It's also compatible with high frequency sampling. Because it's so non-invasive, because you can get it past ethics very easily for sampling quite frequently, you can use it to monitor, for example, drug effects really quite frequently. VOCs are also reflective of what's happening phenotypically. Because they're metabolites in general, they're going to show you what's actively happening in the body and with the disease at any one particular time when you take the sample. And then finally, as I've highlighted on the first slide, this isn't just for respiratory disease. VOCs do perfuse via the blood into the airways from other tissues, so you'll be able to look at this from multiple diseases as well. So what do all of these potential benefits of breath mean for drug development? When I talk to scientists who are planning clinical studies on drugs in development, they generally give me a few different reasons why breath is potentially interesting for them, and I'm just going to talk through some of those. So, for example, one of the things I hear frequently is it offers accessible sampling of the respiratory system. BAL and sputum can be challenging to obtain, and I think are increasingly challenging to obtain. So the potential to obtain something very non-invasively from the respiratory system gives quite nice potential for respiratory diseases. I also hear from people that the ability to kind of frequently monitor patients could be very useful to help them to understand if a drug is having early efficacy signs, or maybe even to monitor patients at frequent intervals to see if adverse events are developing. Because breath has the potential to be developed into something that could be taken, for example, in the patient's home, or at least in, say, pharmacists, this can also be relevant where people want to develop methods to support decentralized trials. The two below identify and non-invasively indicate novel phenotypes of disease and identify novel biomarkers that are kind of related. But this relates to the fact that these VOCs are novel molecules. People haven't generally investigated them before. So they do give you potential to find novel biomarkers that haven't been looked at in the past. For example, if you want to find novel ways to stratify patients or find novel groups of patients who might respond to a particular treatment. And then finally, when people are thinking about post-market drugs, they're often thinking about ways to be able to, for example, indicate which patients might need treating. So I've heard people, for example, talk about maybe being able to monitor patients to see if they're having an exacerbation that might need treatment. Or alternatively, if a drug is known to give significant side effects, I've heard it can be very important to indicate to both the patient and the doctor that the drug is actually doing something to justify those side effects, essentially. And those kind of post-market tests really need to be patient-friendly and non-invasive to motivate people to do them. So hopefully that gives you a few good reasons to listen further. I'm now going to tell you a little bit more about how Alstone can help you to leverage breath to support drug development. But first, I need to tell you a little bit about where breath is right now. As I said earlier, lots of VOCs have been reported to associate to lots of different biological pathways and to different diseases. When it comes to translating this kind of technology, there are actually lots of different options for both breath collection and analysis. Analysis includes platforms like GCMS. And if we consider its sister technology, LCMS systems are already available as IVD medical devices. So it's not too far of a leap to think of something like GCMS being able to be translated for medical device use as well. What we have seen, despite this very promising set of evidence in the literature, is that only a small number of clinical breath tests are actually already in use. For example, there's FeNO for airway inflammation, HMBT for SIBO and carbohydrate malabsorption, hydrogen pylori testing, and methacetin testing for liver function. But that's a relatively small number of tests considering the promising literature. So I guess the question we have to ask is, why don't we see more breath biomarkers already being used in clinics? We think there's a few reasons behind that, and Alstone have basically spent quite a lot of time thinking about the reasons for that and trying to develop something that supports the development of clinically useful biomarkers as efficiently as possible. What we don't want to do is just continue further research that says breath could be a really interesting matrix. It's got potential relevance to lots of different diseases. We want to make sure that when we're doing work in this field, we want to make sure those findings are translatable and we can help translate them. So Where the field is right now, the critical bit for most applications is going to be the identification of suitable VOCs. And that might involve replicating and confirming if VOCs have already been highlighted in the literature, it could be suitable biomarkers, but it could also involve seeing if there are other VOCs. So we do discovery in two broadly different ways. Firstly, from a kind of top-down approach, we take breath samples from humans in human clinical studies and use what we call our breath biopsy Omni platform, which I'll talk about a little bit later. This is an unbiased analysis of a really wide range of VOCs to really open the net wide and see what biology tells us could be relevant biomarkers for a particular application. We can also look at this from a more bottom-up approach focused on specific questions. So, for example, we can do in vitro studies to see if there are VOCs altered when target biological processes and cell lines are activated or inhibited. This might help, for example, to find potential target engagement or efficacy markers. We can also look at more of a pathway analysis approach. So, for example, are there known enzymes involved in a target pathway that could process or potentially produce a volatile that can be monitored in patients' breath? And Tina will give an example of this in the liver disease example later. These findings from these bottom-up approaches can then, of course, be validated in breath using targeted assays and focusing on the molecules that people have identified in these earlier stage approaches. So the key thing to our discovery approaches that makes them a bit different is they focus specifically on identifying VOCs that can be more easily clinically translated and studied to really understand the biology of those molecules. And that's quite different than a lot of other approaches that have been used previously. Once you've identified the molecules, there are then actually multiple options to translate and deploy these technologies. And Alstone as a company is already developing and deploying these kind of approaches. The very easiest option that you can consider is something like central lab-based testing, with breath sampling either performed in the clinical environment or in the home environment. To give you an example of how this is being used right now, we currently provide hydrogen and methane tests for SIBO and food intolerances, where sampling kits are shipped to the patient's home. They breathe into these sampling kits, do the test, and then they send the sample back to our labs just by the post for analysis. We're also, of course, looking at alternative ways of testing patients. So, for example, we're developing point-of-care devices to look at specific molecules, which could be breathed into for very rapid and online analysis. To go into the discovery approaches in a bit more detail, I'll talk about sample collection first. One of the key challenges in finding suitable VOCs in breath has really been how to take a good breath VOC sample that can be reliably compared with another breath sample, either from the same person or from different people, and really focusing here on minimizing the technical variability that's going into a sample. For discovery, there's also the additional challenge of actually how do you ensure that you're analysing what's coming from the patient's breath and not coming from the environment, because VOCs are in the environment around us. To enable this, Alstone has developed this breath biopsy collection station, which is shown on the slide here. This is installed at clinical sites to enable the collection of very high quality breath samples with minimal variability and good signal to noise. It consists of the receiver, which is the piece to actually collect the breath sample. It has a disposable mouthpiece for patient safety. The Casper scrubs the environmental air around the patient and then feeds up that cleaned air to the patient to ensure they're breathing air that's not contaminated with environmental VOCs. And then we've also got a PC which contains our breath collect software. This really automates the process of breath collection. It ensures it's collecting a fixed volume of breath very easily. It ensures you're collecting the optimal breath fraction, but it also generally guides the user towards very successful collection of a good breath sample. So the sampling system is in general designed to be as easy to use as possible, to be as consistent as possible, even between different clinical sites. The samples themselves are collected onto these sorbent tubes that you can see on the top left here. These basically contain a matrix that the VOCs stick to. So as the patient's exhaling into this receiver device, the VOCs from the air are going through these tubes. The air is being expelled. The VOCs are sticking to those sorbent tubes. These sorbent tubes can then just be very easily taken out of the receiver, capped, and then sent to Alstone for analysis at room temperature. We also, of course, support in vitro projects, so where we're looking in vitro to try and find VOCs. Projects of this nature are very much designed in collaboration with customers to ensure that we're planning the experiments to collect the right samples to produce VOCs at the end. In general, we would recommend that the cell or tissue culture is done at the customer site, generally because you've got very experienced people in handling those particular cell lines or tissue culture experiments. That generally gives the best chance of success. The samples would then be frozen, generally snap frozen for shipping to us, so that we can then do the analysis of the VOCs. In general, the sample will need to be incubated and agitated for a little while. We'll work out with you what the most appropriate way to do that is. And that ensures that the VOCs are transferred from the sample into the headspace above the sample. And then those can be absorbed onto these high absorb probes, which contain absorbent material very similar to the absorbent tubes in the previous slide. They can then be analysed by GCMS. For analysis of a VOC sample, we also want to avoid introducing any technical variability into the sample. And we want to ensure that a broad range of VOCs can be analysed. Most critically, though, especially for untargeted discovery analysis, we want to enable the accurate identification of VOCs, such that those VOCs can be further translated and studied with confidence. What we've seen is that too frequently in this field, untargeted discovery studies have shown some really interesting findings. They've shown the presence of some really interesting VOCs or breath patterns, but they've not conclusively identified what those VOCs are, which then makes it really challenging to then further translate those, because you don't know if your targeted assay is indeed looking at the right molecule, and it's very hard to understand biology if you're looking at the wrong molecule too. So we've optimized an untargeted GCMS methodology and QC processes to go alongside this on some very high specification instruments to overcome some of these challenges. And critical to this is development of this growing breath biopsy HRAM library. And these are compounds that we've run on our instruments and can provide high confidence identities when we see these compounds in studies. It's not just the standard way of doing this, which is NIST identification using an external library, which provides much more tentative identification. The HRAM library should provide much higher confidence identification than NIST. So in general, untargeted analysis is what we focused on in Alston. It's generally what's recommended in the VOC space, as it can be used to both replicate previous findings from other studies, but also find additional VOCs with our more advanced platform. However, we can, of course, do targeted analysis. For example, if we think a specific process is hypothesized, we can focus on relevant VOCs, for example, when we're doing in vitro studies. So putting this together, our Omni platform, which I mentioned earlier, is the end-to-end optimized and untargeted biomarker discovery platform, which we use from human breath samples. So this involves breath sample collection with the breath biopsy collection station, shipping of those breath samples in absorbent tubes, room temperature to Alstone. We would then do that untargeted GCMS analysis. And then we'd report at the end on the VOCs that we've identified and provide biological interpretation on how those might be connected into the disease as well. The key things that make this platform different than what's been previously done in the field is, firstly, our focus on enabling that accurate molecular identification with that high resolution accurate mass library. We've also enabled a really wide range of VOCs to be studied with this platform to expand the possibility of finding relevant VOCs. It focuses on measuring the endogenous VOCs. We've done a lot to try and minimize any background incorporation into samples so that we can have higher confidence that the VOCs identified from this platform are actual real biological VOCs. And then finally, the whole platform is optimized end-to-end to minimize any technical variability so that we can really see the biology here. We can see small effect sizes and we can understand if a VOC has suitable natural biological variability to make it a useful biomarker. So just to give an indication of this platform and how it's performing, we typically see about up to 1,500 VOCs in an individual sample. But actually, when we apply our additional criteria, which I'll talk about in a few minutes, only about 500 of those are actually considered as on-breath compounds. So only 500 of each of those are potentially useful biomarkers, we think. When we look at those on-breath compounds across 4 volunteers, we see a median RSD of 26 to 36%, which is reasonable considering we're looking at metabolites here. So I've talked a little bit now about our breath biopsy VOC library. Of course, that's only useful if it's containing VOCs that are potentially good biomarkers. So our VOC library needs to contain VOCs that have a few different properties. First, we need to know and be confident we can see it in our methodology. That's nice and easy to check. We check that we can see the compound using standards. We can see that it absorbs onto the absorbent tubes and the absorbent material we're using. We can check it comes off. And we can check that we can see it in our GCMS methodology reproducibly as well. So everything that's in there will be tested in that way to know that we can see it in our method. Secondly, I've mentioned this in the previous slide, we need to be confident that these molecules are on breath and not from the environment. So other studies out there typically, when they're reporting on their platform performance, typically quote the number of overall detected compounds, which can be very high. But as I showed in the Omni slides, in our experience, quite a lot of the compounds that we see are probably contaminants and don't relate to patient biology. They're not considered on breath. For a VOC to be a suitable biomarker, we need to be actually confident it's coming from breath and not from somewhere else. So, for example, if it's coming from the air the person is inhaling or from any sampling hardware, that's not going to make it a good biomarker. And we define an on-breath compound to be 3 times standard deviation above the background. To ensure our breath biopsy HRAM library is populated with suitable VOCs that we have confidence in could be good biomarkers, we're actually currently doing a project to map and identify VOCs that we consistently see as on breath in a healthy and diverse population. And we're adding to this also with customer studies looking at disease patients. And the VOCs that we're seeing as on breath are being added to this breath biopsy VOC library. So this is an example of what we see and what I'm talking about when we talk about on-breath versus not on-breath compounds. Isoprene is a compound that is very frequently reported to be in breath. And indeed, what we see when we look at a system blank versus breath is that isoprene is at a very much higher level than the system blanks. We also see when we take a double breath sample that isoprene approximately doubles, which is what we'd expect to see if this compound is truly on breath. If we take the example of 1-butanol, though, which has also been reported as a potential VOC biomarker in the field, we can see that in our hands, in this healthy population at least, can see that the compound is not present at a significantly high level on breath versus system blank. And when we take a double breath sample, we can also see that it's not present as a. When we take a double breath sample, we can see that 1-butanol is not doubled. So the other aspect that's important when we consider which VOCs we put into this library is we want to ensure that VOCs are put in that are reflecting relevant and interesting biological processes. So I'll just talk through a few of the processes that we've looked at and thought through that could be generating VOCs that are related to relevant biology. So fatty acid oxidation, for example, produces a variety of different VOCs. And some of these are also reflected in our HRAM library. The gut microbiome, for sure, is going to produce a lot of VOCs that are then reflected in breath. And again, there are some compounds that we can see in that library that we have here as well. Products of amino acid metabolism, so things like tryptophan, phenylalanine, tyrosine, both the buildup of those compounds and the degradation of those compounds and their downstream metabolites are also often volatile and can be seen in breath. And those are in our HRAM library. And then finally, we've got things like lipid peroxidation, where in conditions of oxidative stress, the lipid membranes in cells are broken down, and the breakdown products of those lipids are often volatile, things like hexanal, other aldehydes, and things like that. So finally, the other thing we want to ensure goes into our VOC library is compounds that have been previously identified in other studies, such that we can replicate those findings when we do those. And Tina's now going to talk through a few different examples of places where our technology and others' technology has been deployed to look at VOCs in the context of different diseases. Hand over to you, Tina.
Thank you, Liz. So people with idiopathic pulmonary fibrosis have fibrosis and scarring in the lungs, which not only impacts the function of the airway, but is also prevalent. IPF diagnoses in patients are often delayed due to nonspecific symptoms, resulting with poor prognosis. Therefore, identifying biomarkers with a non-invasive device will be helpful for disease detection. In this study, collaboration with the Three Lakes Foundation and Brigham and Women's Hospital, healthy control, ILA, and IPF subjects were recruited. Breath samples were collected pre- and post 6-minute walk test in all 3 groups, as well as other pulmonary function test parameters such as forced expiratory volume, forced vital capacity, total lung capacity, and diffusion capacity of the lung for carbon monoxide. The goal for this study was to compare biomarkers found in literature, identify novel biomarker candidates that can discriminate IPF from healthy controls, and determine biomarker candidates associated with IPF-related clinical variables. Note that the recruitment of ILA subjects was strongly impacted by the pandemic. Therefore, breath VOC comparisons with the ILA group was expected to be underpowered. Here we show some key results in the study. We identified 22 VOCs significantly different between IPF and healthy controls that were sampled before the 6-minute walk test. One of the VOCs we found was acetoin, a compound used as a flavoring agent in foods. Acetoin was found associated with IPF disease in a published literature, and here we find similar directionality. In addition, 3 VOCs were identified as informative predictors of IPF status. Ethanol 2,4-methoxy-1-phenyl is closely related structurally to acetophenone, which is used as a plasticizer in plastics and concrete and has been identified as a biomarker for lung and breast cancer. 2,3,4-trifluorobenzoic acid, 4-nitrophenyl ester, is a fluorinated compound that is not produced endogenously by humans and might be ingested from fluorinated water, therapeutic agents, and other exposures. These 2 compounds were found positively associated with IPF. Eucalypto is commonly used as a flavoring agent and is a monoterpene which we have confirmed as an on-breath compound. Eucalypto was found negatively associated with IPF. In our study, we measured diffusion capacity of the lung for carbon monoxide to monitor the progression and severity of IPF. 40% of the IPF findings has also showed association with DILCO. Eucalypto, which was one of the 3 informative predictors of IPF status mentioned in the previous slide, was significantly positively associated with DILCO. These results imply that there may be some impact on diffusion of compounds from blood to exhaled breath. In this study, we were able to validate previously reported IPF biomarkers. We were also able to identify new biomarker candidates that differentiate between disease and control groups, which also show statistically significant association with physiological parameters. Our platform and workflow allows definitive identification of compound and subsequent validation. It may be challenging to decide what to do next and move beyond discovery studies. Here, I like to present a good example of how you can design a mechanistic study to develop some understanding of the plausible origins of a biomarker. COPD is characterized by lung tissue damage and reduced lung function resulting from tobacco smoke or air pollutants. The eosinophils activated by stimuli such as interleukin-4, interleukin-5, and interleukin-9 within the lungs can release tissue-damaging granule proteins and oxygen-free radicals, which together orchestrate bronchial inflammation and cause severe damage to the lung epithelium. The chronic disease is the 3rd leading cause of death worldwide, causing 3.23 million deaths in 2019. In this study, a biological treatment was aimed at reducing lung inflammation in COPD subjects. 23 COPD subjects were each given one of 4 treatment regimes, placebo or one of 3 drug doses. Subjects were each given 3 treatment doses between visits 2 and 7 of the experiment. Largely, subjects gave 2 breath samples pre-treatment and 4 around treatment. The primary goal of this study is to identify biomarkers that are associated with response to treatment and stratify responders. The secondary goal of this study is to understand the associations with inflammation markers. The key results of this study is that we found one VOC compound, molecular feature 136, which is classified as an alcohol, correlated significantly with blood eosinophils at pre-treatment baseline. The compound also tracks with changes in blood eosinophil over time. The inflammatory process can produce reactive oxygen species, which can react with fatty acids and/or phospholipids to generate volatile biomarkers on breath. Molecular feature 136 could be a product from lipid peroxidation due to interaction between ROS and unsaturated lipids. In a separate study, it showed that in vitro activation of eosinophils using PMA produced the same compound. Prediction of eosinophil counts showed the fitted values from the subject and visit fixed effects model for molecular feature 136 are close to the line of agreement when plotted against the observed eosinophil counts. Although within individuals, changes to blood eosinophils were relatively small in this study, molecular feature 136 correlated with blood eosinophil levels both cross-sectionally and longitudinally, showing it is a promising candidate biomarker for treatment to reduce lung inflammation in COPD patients. The study demonstrates how our platform can help develop some understanding of the plausible origins of a biomarker. Aside from identifying potential biomarkers for respiratory diseases, breath biopsy also offers the opportunity to collect a sample that is representative of tissues throughout the whole body. The lung alveoli are structured to enable high-efficiency exchange of VOCs between breath and blood, and the circulation of all blood through the lungs is about once per minute, enabling assessment of systemic disease information. The liver, for example, is an ideal target for breath profiling due to its rich blood supply and high metabolic activity. Over 30% of adults in some countries have chronic liver disease, yet more than 50% are only detected in the later stages. With the current gold standard invasive tissue biopsy, which is a painful and difficult method for disease detection, breath biomarkers could be an effective option for early-stage screening of at-risk individuals. The potential of VOCs in breath as biomarkers for cirrhosis has been shown in published literature. In the study, healthy subjects and those with liver disease were recruited, and 12 of the subjects' breath samples were taken before and after liver transplant. The study found that limonene, which is a terpene with exogenous sources, such as ingestion with fruits, beverages, and flavored food in our diet, increased in liver cirrhosis patients due to its reduced liver enzyme activity. The limonene levels were able to return to normal after liver transplant. Most of the drugs are metabolized in the liver by only 4 enzymes that belong to the CYP450 system. The CYP2C family specializes in metabolism of a variety of compounds, including drugs like celecoxib and warfarin, endogenous compounds like serotonin, and dietary compounds such as limonene. In the presence of liver cirrhosis, CYP2C9 and C19 activity are downregulated, leading to the accumulation of limonene in the adipose tissue. Gradual release from the adipose tissue results in prolonged permanence of limonene in circulation, leading to increased secretion in breath. In our in-health pilot study with 40 healthy controls and 44 subjects with cirrhosis, we did a targeted analysis of exhaled limonene, and our study supported the published literature, which showed limonene increased in subjects with cirrhosis, and the concentration of limonene was correlated with disease severity, which was assessed with Child-Pugh classifications. The discrimination model showed 0.73 and 0.77 of sensitivity and specificity respectively. In addition, we also found limonene correlated with blood biomarkers of liver function but not liver damage. With our breath biopsy Omni assay platform, we performed another in-house study, recruiting 42 healthy controls and 46 subjects with cirrhosis for untargeted analysis. In this study, 196 on-breath VOCs were detected consistently across samples, and 29 of them were significantly different between healthy controls and cirrhosis samples. This included limonene, which confirms our result from previous targeted analysis as a relevant biomarker for chronic liver disease as well as other studies. Currently, we have confirmed several on-breath compounds classified as terpenes in our Omni platform. These terpenes are likely from plant dietary origins. Limonene in our liver disease study, as well as eucalypto in the IPF study mentioned earlier, are both confirmed as on-breath compounds in our platform. These compounds are generated in plants either from pyruvate through the MEP pathway to produce monoterpenes, or from acetyl-CoA through the NVA pathway to produce cis-quaterpenes. Other terpenes that have been suggested to associate with disease in published literature, such as alpha-pionein and 3-carine, have also been confirmed as on-breath compounds in our Omni platform. To date, the understanding of which CYP enzymes are involved in these different terpenes and what metabolites are being produced downstream are limited to only a few in vitro in vivo studies and in silico predictions. And as mentioned earlier, in vitro studies have suggested limonene metabolized by CYP2C9 and CYP2C19. Other top VOCs that were significantly different between cirrhosis and healthy subjects include dimethylselenide and 2-pentanone. Models built on these compounds individually show that they are decent predictors for cirrhosis. Dimethylselenide is one of the excretion products of selenium metabolism. The downregulation of dimethylselenide in the breath of cirrhotic subjects is likely due to impaired organic and inorganic selenium biotransformation. 2-pentanone is a ketone we have confirmed as an on-breath VOC. 2-pentanone is also the compound with the highest confidence for disease prediction in this study, with an area under curve at 0.82. The increased level of 2-pentanone in cirrhotic subjects is possibly due to increased incomplete oxidation of fatty acids. Fatty acids are an important energy source. The abundance can fluctuate with circadian rhythms and energy demands. Oxidation of fatty acid is a cyclical process, where acetyl-CoA, a 2-carbon unit, is broken off from acyl-CoA repeatedly until the precursor is completely oxidized. This process is highly efficient, and intermediates of fatty acid oxidation are generally completely metabolized. However, volatile ketones such as 2-pentanone may arise through incomplete oxidation. The 2-carbon unit, acetyl-CoA itself, can be processed into several volatile molecules, such as acetic acid through hydrolysis, acetaldehyde through acetaldehyde dehydrogenase, and acetone through carboxylation. These molecules are all found on breath in our dataset. We also compare blood biomarkers of liver function, albumin, bilirubin, and INR with our top 11 VOC compounds. And these 11 VOC compounds collectively show correlation with measured blood biomarkers. Limiting correlations are also consistent with those measured in our previous study. In addition, PCA plots based on these 11 VOCs show the ability to separate Child-Pugh scores above 5 from healthy controls, suggesting that VOCs correlate with disease severity and therefore may be relevant for early detection. Overall, the VOCs correlated well with clinical measurements of liver disease progression. Our study shows that the breath biopsy Omni is not only a reliable platform for supporting disease-associated VOCs attained through other methods in previous work, but also able for new biomarker discovery. The newly identified VOCs can be used to produce an effective model for detecting cirrhosis, and our top VOCs correlated well with the clinical measurements of liver disease progression.
Thanks, Gina. So to summarize, Alstone Medical is in this space to help people to work out if there are breath biomarkers that could be useful to help them in their drug discovery processes. We have developed really robust breath sample collection technology to enable samples to be collected that can be compared reliably between different samples, and also capture a sample with a very high signal-to-noise ratio. We can also do in vitro identification of relevant VOCs and support with customers to be able to capture VOCs produced from in vitro work. Those VOCs, and we've developed various processes to analyze those, and we can analyze those via high-spec GCMS to identify specifically which molecules are potential good biomarkers, so that those can be translated to further clinical use. And then, of course, in the future, once we have identified the relevant markers that we're confident in or good, useful biomarkers, we can support in the translation of those VOCs to further fit-for-purpose assays and tests. I'm going to stop there. If anyone has any questions, very happy to take those. I hope that was a useful introduction for you.
So thank you very much, Liz and Tina, for that interesting presentation. I would now like to open the floor for discussion. So remember to send us any of your questions using the question panel on the platform. So our first question today comes from Amina, who's a resident doctor in Spain, and she would like to know what non-invasive biomarkers you would recommend in both obstructive sleep apnea and primary pulmonary hypertension.
There have been some studies in both disease areas, and it's suggested that depending on the sleep cycle, there may be reduced fatty acid oxidation and also elevation on the TCA cycle intermediates in sleep apnea. For PAH, the VOC acetaldehyde has been discovered to increase on breath in disease subjects, which can probably originate from oxidation of ethanol, which is produced by the gut microflora. Other biomarkers such as pentane or alkanes and alcohols has also been identified to increase in these subjects, which could suggest inflammation or excessive proliferation associated with local hypoxia. So we will recommend approach that is able to monitor these VOCs specifically, but also we'll suggest to look more broadly using an untargeted method. In addition to validating these existing markers, there may be additional VOCs that can be better either alone or together as a panel.
Great, thank you. So our second question comes from Dr. Tai in Vietnam, who asks, Pheno requires different expiratory flow maneuvers in order to reflect different parts of the airways. Is this the same as VOCs?
Thank you. That's a really good question. When we're looking at VOCs, we're actually just taking normal tidal breathing. So the patient would just breathe into our device for a period of time, just breathing as they normally would. And the device itself is basically monitoring how they're breathing so that they can basically fractionate the breath to the appropriate points. Normally what we're looking at when we're collecting the breath sample is we want to collect the lower breath fraction because that contains the most signal and the least noise. And the receiver and the technology we have actually allows them to do that specifically without the patient having to change how they're breathing. I hope that answers your question.
Yeah, that's great. Thanks, Liz. So then we had two quite similar questions. The first one is there any VOC data differences between ethnics? And then the other question that came through, which was similar, is how would diversity in an ethnic sense, i.e. relating to culture, etc., affect VOC differences between groups of different ancestry? And then in brackets, I assume that the experiments at Brigham mainly were done in white subjects.
So in the IPF study, the main subjects were indeed Caucasian. So we have been trying to confirm VOCs that are on breath in Owlstone, and when we designed it, we did consider the ethnicity which could affect the differences of VOC. So when we recruited the subjects, we had recruited the ethnicities in proportion with the same ratio as the UK populations.
Great, thank you. And then another question that we had come through from the audience are how many VOCs can be identified in a non-biased approach?
Yeah, another great question there. It's a really pertinent one when we're thinking about, especially unbiased approaches, because obviously the more molecules you can analyze, the better, really. But there's also the question of making sure you're analyzing the good molecules. So in our Omni platform, which is a untargeted platform, on a kind of typical sample, we see somewhere around about 1,500 individual VOCs. But actually, when we apply additional criteria onto that, as in which of those VOCs do we actually see as on breath and above background levels. And that drops down to about 500, but that's 500 VOCs we're confident could be useful molecules to study. So there's quite a large range of diversity there that you can look at with breath.
Great, thanks, Liz. And now just coming on to the last question. So how broad are the applications of breath testing?
That's probably one for me again. I'd say really the limit is your imagination. There's been a wide range of studies that have been previously published in breath, looking at a huge different range of diseases, and a lot of them have found differences between disease patients and healthy controls, for example. And there are lots of different reasons why biologically that might be the case. So, I mean, the cases, I guess, where we see most interest in looking at breath are more about where something that's non-invasive is really, really useful. So, for example, when someone wants to be able to monitor patients after, say, a drug has been given very frequently at very frequent intervals, so they can see at an earlier stage, actually, is this drug doing something? Can I just stop this study a little bit early and save a lot of money, for example? In terms of the therapeutic areas that we see where this is really seen as potentially useful, respiratory is certainly one. We know that a lot of the kind of ways to look at the lung specifically, things like using BAL, things like using sputum, can be quite challenging to get, especially in drug trials. And so breath VOCs provide another way of looking at kind of lung-generated molecules that could also be relevant to the disease biology, but without requiring that kind of invasive or challenging to get sample.
Great, thank you so much, Liz. And thank you to both Liz and Tina for answering the questions and for your presentation. So that draws the webinar to an end. Just a reminder that a full recording of this session will be made available to download on our secured website, and we'll send you the details for this in a follow-up email. So yes, thank you again for everyone's questions and for attending this webinar. For further information regarding Oxford Global's Biomarker series, please visit oxfordglobal.co.uk/biomarkers. And all that remains to be said is have a great day, and we look forward to welcoming you to one of our future events.